Hello, this is Yunha, and as introduced, I am still currently a postdoctoral fellow at the Rockefeller University. And today I'd like to introduce some of my recent findings, which is about a tissue injury sensing and repair pathway that we discovered. So the maintaining homeostasis is a hallmark of biological system. Therefore, the living organisms try to restore it as soon as possible when the homeostasis is disrupted. A representative example of the disruption of homeostasis is pathogen infection. Once the tissue is infected by pathogen, it senses the non-self patterns such as bacterial DNA or viral RNA, which can induce the production of interferon, whose downstream pathway involves the activation of STAT1 and 2 to defend the host against the pathogen. Another good example of the insult that can disrupt the homeostasis is tissue injury. However, due to its complexity, how the tissue senses and responds to the injury is still under the process of investigation. So the question we wanted to answer was, how does epithelial tissue sense and respond to the tissue injury? To answer this question, we used the mouse skin as a model system. These are the very brief schematics of the skin epithelium tissue structures, as well as the process of injury, repair, and the final wound closure. The outermost layer of skin is referred to as epidermis, and in its basal layer, there exist the epidermal stem cells, which are the tissue-resident stem cell population in the skin epithelium. The underlying layer is called dermis, and there exists a variety of different cell types in this layer, including the endothelial cells that consist the blood vessels, immune cells such as macrophages, or the fibroblasts, which produce the extracellular matrix. Once the tissue is injured, one essential step that has to happen for the tissue to be repaired properly is the reepithelialization. Reepithelialization is governed mainly by the epidermal stem cells, where these stem cells proliferate and migrate to finally close the wound. Previously, it has been shown that STAT3 is significantly activated in these epidermal stem cells following the injury, and that such activation is essential for the reepithelialization to properly happen. We got the hint from this previous finding and first wanted to figure out the upstream cue that can cause the STAT3 activation in the stem cells during the injury. So this is the approach that we took to get the answer. We took the list of the cytokines that are known to activate STAT3 and performed a quantitative RT-PCR on the dermis as well as in the epidermis, in the homeostatic skin without a wound, and also after wounding the tissue. And as you can see in this figure, we discovered that IL-24 or interleukin-24 is significantly and specifically upregulated in the epidermis only following the injury. To further specify the source of this wound-induced IL-24, we sorted out different cell types in the skin, including the epidermal stem cells, fibroblasts, endothelial cells, and immune cells, and again performed a quantitative RT-PCR to look into the IL-24 level after wounding the tissue, and discovered that it was epidermal stem cells that produced the IL-24 following the injury. This was further confirmed using the single-cell RNA sequencing, since we performed the 10x RNA sequencing in the wounded tissue, where we discovered that it was indeed the epidermal stem cells, specifically located on the wound edge, that produced IL-24 when the tissue was injured. To give you very brief information of IL-24, IL-24 or interleukin-24 belongs to IL-10 cytokine families with IL-10, IL-20, IL-19, or IL-22. But unlike some of the members in this family, the role of IL-24 in the context of wound repair has been still controversial, since some say that it's for the tissue repair, while the other says that it's against it. But interestingly, we discovered using the phylogenetic analysis that IL-24 and its receptors that we think would be involved in the tissue repair are homologous to the interferon and interferon receptors, if you remember from the introduction slide, which are well known to be involved in the pathogen defense. In figure 8, the yellow and orange areas respectively show the IL-10 cytokine family members and interferon family members, which share a very close homology during the evolution. And likewise, the figure B shows with the blue and green areas, the long and short subunits of the interferon receptor family members and IL-10 cytokine receptor family members, which again share a very close homology. So this piece of information suggests that IL-24, which again we think would be involved in the tissue repair, and the interferon pathways, which are known to be involved in the pathogen defense, are likely to share a common ancestral signaling. In other words, these two pathways might have shared the common ancestor and might have been bifurcated at some point during the evolution so that they can defend the host more effectively in a more specialized way against the insults that can disrupt the homeostasis, whether it's tissue injury or pathogen infection. So based on the information, this was the hypothesis that we set. Given the homology shared between the interferon and IL-24, as shown by the phylogenetic analysis, as well as the homology shared between the STAT1 and 2 and 3, since they are in the same STAT family members, we hypothesized that there exist the tissue injury pathways that is parallel to the pathogen infection pathways, where the epidermal stem cell-induced IL-24 play an important role for the repair. Now that we have the hypothesis, we had to test whether the IL-24 is indeed playing a role in the tissue repair. As you can imagine, there are so many different cell types in the tissue epithelium. So therefore, it was almost impossible to test the behavior of every single cell types in the skin. Therefore, we decided to focus on the cell types that have a higher potential to react on the IL-24. For that, what we did was we looked into the levels of IL-24 receptors in different cell types and focused on the cell types that have the higher levels of IL-24 receptors, or I would say just a decent or significant detectable level of IL-24 receptor. With this approach, we found the three cell types would be the one that we should focus on. Specifically, first, the epidermal stem cells, which are the main source of IL-24. Secondly, the endothelial cells that comprise the blood vessels. And thirdly, the fibroblast. While focusing on these three cell types, we performed the punch wound, thickness punch wound in the back skin of wild type versus IL-24 knockout mice to test the role of IL-24. The images that I'm going to show you in the later slides are the sagittal view of day 5 post-wounding. First, we looked into the response of epidermal stem cells, which are located in the basal layer of epidermis, which was stained with its marker keratin 14 and is shown with a green color in this image. The upper image is the wounded tissue from the wild-type mouse, and the lower one is the one from IL-24 knockout mouse. Among various cell behavior, we specifically looked into the proliferation of these epidermal stem cells using the EDU pulse, which is known to be incorporated into the proliferating cells and shown with the white signal in this image. If you focus on this magnified inset, upon wounding, the epidermal stem cells are actively proliferating as shown with the EDU staining to mediate the reepithelialization to finally close the wound. However, at the same time point in IL-24 knockout mice, I hope you can appreciate that the percentage of the proliferating cells is greatly reduced. indicating that the reepithelialization is impaired. Then we also looked into the response of endothelial cells using its marker CD31. These are the same images, but now let's focus on CD31, the marker for endothelial cells that can generate the blood vessels, which is shown with the purple color. In the wild-type mouse, the majority of the dermal wound bed is covered with CD31 staining, suggesting that the blood vessel is actively regenerating so that it can support the tissue regeneration by providing the oxygen and the nutrient that is necessary for dermal repair, as well as the reepithelialization. But in IL-24 knockout mice, there's a clear lack of CD31 staining, showing that the blood vessel is not properly regenerating. In addition to staining the tissue section, we also performed the whole mount staining to have a better idea about the response of endothelial cells. Here, the epidermis is stained with the keratin 14, the green color, and the endothelial cells are stained with endomucin, the red color. Very consistently, in the wild-type mouse, the endothelial cells are actively regenerating the blood vessel and is recruited near to the epidermis so that it can support the reepithelialization. But in IL-24 knockout mice, the lack of overlapping signal between the epidermis and endothelial cells suggests that the recruitment of the endothelial cells near to the epidermis is impaired in this case. Lastly, we looked into the response of the fibroblast using its marker PDGFR alpha, the green signal in this image. And just like what we did for epidermal stem cells, we tested its proliferation using the proliferation marker Ki67, the white signal. Very consistent to what was observed in other cell types, in contrast to the wild-type mouse where the fibroblasts are actively proliferating, as shown with the co-staining of the Ki67 and PDGFR alpha. In IL-24 knockout mice, the cells are not actively proliferating anymore. In addition to the proliferation of the fibroblasts, the very important role of fibroblasts during the tissue injury and repair is to produce the collagen. The collagen, more specifically collagen 1, is one of the extracellular matrix that is known to be produced by the fibroblast during the tissue repair so that it can support directly the dermal repair as well as indirectly the reepithelialization. So to test this ability of fibroblasts to generate the collagen, we stained the tissue with the collagen 1, shown with the purple color in this image. Very consistently in wild-type mouse, where the majority of the dermal bed is covered with collagen 1, showing that the fibroblasts are actively producing this extracellular matrix. But in IL-24 knockout mice, the yellow asterisk shows that the ability of the collagen 1 production of fibroblast is greatly impaired. So to briefly sum up the things that I showed you so far, I showed you that upon the wounding, the epidermal stem cells produce IL-24, which can act on various cell types to coordinate the tissue repair. First, it acts on the epidermal stem cells in an autocrine manner to mediate the reepithelialization, but also in the paracrine manner, it acts on the endothelial cells and fibroblasts to mediate the angiogenesis and collagen deposition. Since now we have some good ideas about what is the downstream of IL-24, we wanted to go back up and figure out the non-homeostatic patterns that can cause as the upstream cue to induce IL-24 in the context of tissue injury. As you can imagine, when the tissue is injured, there could be so many different changes that can happen. But since the most prominent impairment and changes that we observed in IL-24 knockout mice was the impairment in the angiogenesis, we posited that the severed blood vessel followed by the tissue hypoxia could be the top candidate. Here, this is the image of the wounded tissue in wild-type mouse, day 3 post-wounding, as opposed to the day 5 wound that I showed you in the earlier slides. For your reference, even in the wild-type mouse, in day 3, it's yet for the blood vessels to be fully regenerated. We stained this tissue with the pimanidazole, the hypoxic condition marker probe, and stained it with the green color in this image. And I hope you can appreciate that upon the wounding, a majority of the dermal area near the epidermis, as well as the wounded epidermal area, are under severe hypoxic condition. In addition, we also discover that the epidermal stem cells located in the epidermis here exhibit a greatly upregulated level of HIF-1 alpha at this time point. So HIF-1 alpha is the transcription factor that is known to be stabilized and upregulated during the hypoxic condition. And all the more interestingly, when we followed up to day 5, where the blood vessel regenerates in wild-type mouse, you can see that the epidermal stem cells here are provided with the oxygen by the newly regenerated blood vessel and lost their HIF-1 alpha expression. Whereas in IL-24 knockout mice where the angiogenesis is impaired, the HIF-1 alpha staining is maintained longer as the epidermal stem cells cannot be provided with enough oxygen yet. So these pieces of information suggested and gave us some hint that there could be the potential correlation between IL-24, angiogenesis, HIF-1 alpha, and hypoxic condition. Therefore, to test whether the HIF-1 alpha could really indeed serve as the upstream cue to induce IL-24 during the injury, we took advantage of the HIF-1 alpha conditional knockout mice. Specifically, we generated this line by breeding the keratin 14 Cre-ER mouse to specifically target the keratin 14 positive epidermis with the HIF-1 alpha flux line so that we can generate the HIF-1 alpha conditional knockout line. We again did additional crossbreeding with this line with the YFP fluxed mice line so that we can tag the HIF-1 alpha conditional cells with YFP. After topically treating this line with the tamoxifen to activate the Cre-ER, we wounded the skin and again sorted the epidermal stem cells and measured the IL-24 level. Here, as mentioned, YFP negative cells are the wild type epidermal stem cell, and the YP plus cells are the HIF-1 alpha conditional knockout cells. The left side is under the homeostatic condition, and right side is the wounded condition. As mentioned so far, in the wild-type epidermal stem cells, the IL-24 is significantly induced upon the wounding. But if you compare the induction in wild-type stem cells versus HIF-1 alpha conditional knockout cells, I hope you can really appreciate that there is a great reduction in IL-24 induction. This suggests that even if the hypoxic condition and HIF-1 alpha might not be the only cue to induce IL-24 following injury, it could be one of the very important cues that cause the IL-24 induction. In addition to hypoxia and followed upregulation of HIF-1 alpha, we also wanted to test whether the STAT3 could be another cue to induce IL-24. If you remember this axis, actually the STAT3 is well known downstream of IL-24, not the upstream. But in the pathogen infection pathway that we think would be parallel to our tissue injury pathway, step 1 and 2 not only serve as a downstream of interferon, but also can be the upstream cue to cause the induction of interferon, forming the feedback loop. Therefore, we wanted to test whether this is the case in our tissue injury pathways as well. So to test this idea, we took advantage of STAT3 conditional knockout mice that we generated by breeding the keratin 14 Cre mice with STAT3 fluxed mice. Very consistent to what was observed in the earlier slide in HIF-1 alpha conditional knockout. You can see that the IL-24 induction upon wounding is also greatly reduced and impaired in STAT3 conditional knockout mice, suggesting that the STAT3 not only serve as the downstream, but also can be the upstream of IL-24, forming the feedback loop that is reminiscent of what was observed in the pathogen infection pathways. So such dual dependency of IL-24 induction during injury on both hypoxia and HIF-1 alpha, as well as IL-24 and STAT3, can ensure the fine-tuning of IL-24 to happen only following the injury and reserved after the injury is repaired. So such dual dependency could be further confirmed using the sequencing analysis, as we performed various sequencing analysis to look into the wound-induced dynamics of transcription using the RNA sequencing, as well as the chromatin accessibility and binding using the ATAC sequencing and cut-and-run sequencing at the IL-24 locus. So these 5 red boxes indicate the chromatin region that were opened upon wounding at IL-24 locus, as shown with the ATAC sequencing data. And we discovered using the cut-and-run sequencing that HIF-1 alpha and STAT3 bound to their cognate sites only under hypoxic condition. If you look into this data comparing the normoxia and hypoxia condition. So again mentioned, such dual dependency of IL-24 induction in both HIF-1 alpha and STAT3 can ensure the fine-tuning of such induction. So to summarize, I hope that I could show you today that we discovered the tissue injury pathway that is parallel to the pathogen infection pathway, where the epidermal stem cells sense the wound-induced hypoxia and HIF-1 alpha upregulation to induce IL-24. So this IL-24 can act on different cell types to coordinate the tissue repair, more specifically on the epidermal stem cells to mediate the reepithelialization, but also in a paracrine manner on the endothelial cells and fibroblasts for angiogenesis and collagen deposition. In a direct way, the study, I expect this study to help us to broaden our understanding of how the tissue senses and responds to the tissue injury. But beyond that, given that the serious infectious diseases are often followed by the tissue injury, I hope this study has the potential to provide the therapeutic avenue that is essential for disease tolerance and host survival. With that, I would like to thank my supervisor, Dr. Elaine Fuchs at the Rockefeller University, and my collaborator, Dr. Suchi Liu, who's the co-first author of this paper and worked with me and is currently a professor in UT Southwestern. And I would like to also thank all the members in the Fuchs Laboratory, as well as my funding source during the study, Immuno-Oncology Fellowship. And just add the side note as introduced, the study has been conducted while I was a postdoctoral fellow at the Rockefeller University, and technically I'm still a fellow until today. But luckily, I got a very good chance to have my independent research group at postdoc in the Department of Life Science from upcoming February. So there, based on my expertise in stem cell biology, developmental biology, and intercellular communication, I'm interested in studying the cellular senescence in the context of aging and development using the tissue injury and the mammalian embryo as a model system. So if you're interested in any of the topics that I just briefly mentioned, or the topics that I introduced during today's talk, please feel free to reach out, and I'd be really happy to interact with you. With that, I'd like to thank you for listening, and I'd be happy to take questions. Thank you.